Bioelectronic devices, from electrodes that stimulate the brain to wires that send sound signals to the auditory nerve, have revolutionized the treatment of many medical conditions. In cochlear implants, metal wires encased in silicone are fed through the inner ear to carry electrical signals from a transmitter. But the insertion of wires like these doesn't always mesh well with our body's soft tissue, sometimes causing inflammation or damage to nearby tissue and neurons. Researchers in Sweden explored an alternative to inserting wires by forming gel electrodes in living tissues, which they initially demonstrated in plants. First, researchers tried creating these electrodes in the xylem, which transports water and nutrients throughout plants. They initially tried injecting a gel polymer, a long chain of molecules made up of smaller building blocks called monomers. They hoped the liquid polymer would move through the transport system of the plant, but they found it could only travel very short distances, clogging the vascular system. When they changed the formulation to be made of monomers, the gel moved longer distances and underwent an unexpected transformation. The small molecules in the gel joined together, a process known as polymerization. The bond between these molecules fixed the gel into place and allowed for electrons to flow through, creating a soft conducting wire in the plant. Most soft electrodes grown in living tissue required external electrical or chemical energy to polymerize, which could cause scarring. In this approach, metabolites, small molecules made during a variety of living processes, like energy production, react with an enzyme the plant produces to kickstart polymerization. The next step was perfecting this approach in animals. The ingredients that initiated polymerization in plants are not produced naturally in most animal tissues, so the researchers had to make the process work with other metabolites. In the brain of zebrafish, lactate was an abundant metabolite. In other tissues, like the zebrafish heart, both glucose and lactate were present. For each tissue type, the researchers added in an enzyme that would break down the available metabolite into hydrogen peroxide. Another enzyme reacts with the hydrogen peroxide, causing the monomers to link together, just like in the plant model. This made a gel wire that could successfully carry a current. Because the injected gel is so thin, it can fill in between existing structures before locking into place. This soft wire didn't appear to alter behavior or impact the growth of surrounding cells. In the future, the researchers hope to test this approach on living mammals like mice, and fine-tune the gel to grow in more specific places, perhaps triggered by metabolites related to diseases. It's only a matter of time before wires and probes start to mesh more seamlessly with our bodies.